Hey everyone, so this video is about chapter 16, which is about the molecular basis of inheritance. So this video is going to go into Watson and Crick some, so let's get there. So we have a researcher, his name was Thomas Hunt Morgan. He and his research group, they discovered that genes are located on chromosomes. And remember that chromosomes are the structures inside of cells that carry our genetic information. They also said that chromosomes are made up of two main things, which are DNA, so deoxyribonucleic acid, and proteins. And both proteins and DNA were considered as possible genetic material. So after Morgan's discovery, scientists, they wanted to figure out which part of the chromosome was responsible for carrying genetic information. And they called it the genetic material. And it was either between DNA or proteins. And up until the 1940s, most scientists thought that proteins were the genetic material. And this is because proteins, they are made up of the 20 different amino acids, which means that it allows them to have great diversity in their structure and function. But DNA, on the other hand, it seemed too simple to be genetic material. It only contained 4 nucleotide bases, and those bases, they would have to account for the entire complexity of life. So scientists, they thought that DNA was too simple to be genetic material, and they thought that it was proteins. So this view, it was not challenged until there were certain experiments performed. So in 1928, there was this scientist named Frederick Griffith, and what he did was discover that the genetic role of DNA. And what he did was have a study. He took Streptococcus pneumoniae. It's a bacteria that causes pneumonia in animals, well, specifically in mammals. So he took one strain of it that was harmless to the animal, and he took another strain that was pathogenic. Pathogenic just means that it is disease-causing. What he did was he mixed a heat-killed pathogenic strain of bacteria with a live harmless strain and then injected it into the mouse. And what happened is that mouse died. So this led him to his phenomenon of transformation. He said it was a phenomenon that was now, and it's now defined as a change in genotype and phenotype due to the assimilation of external DNA by a cell. And what this means is that genetic information can be transferred between cells. So the way that this non-pathogenic, this harmless bacteria was able to kill the mouse was because that virulent, that pathogenic bacteria transferred its DNA to that harmless one, which ended up killing the mouse. After Griffith came Avery, McCarty, and McLeod. And what they did was that they focused on the 3 main candidates as the transforming substance of DNA. Those 3 main candidates were DNA, RNA, and protein. Avery, he broke open the heat-killed pathogenic bacteria, and then he extracted all of the cellular components. He then used specific treatments that inactivated each of the 3 types of molecules and tested the ability of each sample to transform harmless bacteria. And DNA was the only one that showed transformation. So then in 1944, Avery, McCarty, and McLeod, they announced that the transforming substance was DNA. And a lot of biologists at this point, they were skeptical because there seemed to be better candidates. And it was also thought that the genes of bacteria, they could not be similar in composition and function to those of more complex organisms. So We ended up getting further evidence that DNA was a genetic material, and this came from studies that tracked the infection of bacteria by viruses, which are called bacteriophages, or phages for short. So, viruses, they consist mostly of DNA enclosed in a protective coat of protein. Sometimes viruses consist of RNA, but in order to replicate, a virus has to infect a host cell and take over that cell's metabolic machinery. And what ended up happening is that in 1952, we had Alfred Hershey and Martha Chase show that DNA was a genetic material of the phage T2. Essentially, what they did is that they took a bacteriophage, they coated its protein, they radiolabeled the protein coat. When that bacteriophage infected the cell, no radioactivity entered. When they grew the phage, they put it through a centrifuge, and when the supernatant, when it separated, there was radioactivity in it. And that all told us that proteins are not genetic material. The next thing that they did was they radiolabeled the phage's DNA. When bacteria was infected, radioactivity entered the cell. When they grew it in the lab, they centrifuged it, they found that there was radioactivity in the pellet. And what this meant was that DNA is the genetic material. So by 1947, there was a scientist named Erwin Chargaff, and he developed the base pairing rules. So basically, he developed a series of series of rules based on a survey of DNA composition in organisms. He knew that DNA was a polymer of nucleotides that consisted of nitrogenous bases, deoxyribose, and a phosphate group. He knew that the bases could be adenine, thymine, guanine, or cytosine. And he also said that DNA varied from species to species. And Chargaff, he found this really peculiar regularity in the ratio of nucleotide bases, and he named them Chargaff's rule. What he said is that in any one species, the 4 bases are found in characteristic but not necessarily equal ratios. And what this meant was that the amount of adenine is about equal to the amount of thymine, and the amount of guanine is equal to the amount of cytosine. However, the amount of purine is not equal to the amount of pyrimidine. He also said that in all organisms, the number of adenines is approximately equal to the number of thymines, and the number of guanines is approximately equal to the number of cytosines. And the basis for these rules, they remained completely unexplained until we were able to discover the double helix. So by the early 1950s, there was a challenge to determine how the structure of DNA accounted for its role in inheritance. And there were scientists that were working on the problem. But it wasn't until Maurice Wilkins and Rosalind Franklin used X-ray crystallography to study the structure of DNA. Using X-ray crystallography, we have X-rays that are going to be diffracted as they pass through specific aligned fibers of purified DNA. The diffraction pattern can then be used to deduce the 3-dimensional shape of the molecules. So what happened is that Wilkins and Franklin, they took this really famous photo 51, which is this photo, and when they saw that X shape, they knew that that heavily suggested a helix. So after this, James Watson, he learned from that research that DNA is helical, and he obtained data and that was got by Franklin and others that were able to suggest the width of the helix and the spacing of the nitrogenous bases. So the width of the helix suggested that it was made up of 2 strands contrary to a three-stranded model that Linus Pauling had recently proposed. So, Watson and Francis Crick began to work on a model of DNA that had two strands, which was known as the double helix. He used unpublished work from Franklin, which showed the backbone was on the outside and the width of the double helix as well. Using molecular models made of wire, they placed the sugar phosphate chains on the outside and the nitrogenous bases on the inside of the double helix. What this arrangement did was it placed our relatively hydrophobic bases away from the surrounding aqueous solution. And in their model, the two sugar phosphate backbones were anti-parallel to each other, with the subunits running in opposite directions. So overall, Watson and Crick, they determined that the chemical side groups of the nitrogenous bases formed hydrogen bonds and connected the two strands. The base pairing rules dictated the combinations of nitrogenous bases that form the rungs of DNA, and that the rules do not restrict the sequence of nucleotides along each DNA strand. And in April of 1953, Watson and Crick published a succinct one-page paper in Nature reporting their double helix model of DNA. And the beauty of this model was that the structure of DNA suggested that basic mechanism of its replication. So eventually, Watson and Crick, they obtained more information that allowed them to correct the double helix. They had originally hypothesized this is what it was like, and after they learned about that specific pairing of nitrogenous bases in DNA, they realized that it was actually a double helix. And that was a possible mechanism for the next step, for the accurate replication of DNA. It was so clear to Watson and Crick from the corrected double helix model. So Watson and Crick, they have hypothesized a semiconservative model for DNA replication. And what that predicted was that when a double helix replicated, each of the daughter molecules has one old strand and one newly made strand. At the same time, there were two other competing models. There was a conservative model in which the two parental strands came back together after the process, and then there was a dispersive model. The idea that all four strands of DNA following replication had a mix of old and new. It wasn't until the late 1950s that these two researchers named Matthew Meselson and Franklin Stahl that they proved the semi-conservative model as correct. What they did was they labeled nucleotides of a strand and separated replicated strands. When they traced what happened, the results matched that of that semi-conservative model, in which each strand has one parent cell and one new strand, not cell, sorry. Each DNA has one parent strand and one new strand. So, DNA synthesis, it is the process by which a cell duplicates its DNA before cell division unwinding. In the S phase of interphase, we have the replication of DNA, and that happens in the nucleus of eukaryotes. So DNA replication will only take place in the S phase, which only occurs during interphase of the cell cycle. So the replication of a DNA molecule, it begins at this origin of replication. In eukaryotes, we might have thousands or hundreds of origins of replication sites per chromosomes. At the origin sites, the DNA strands, they are going to separate. And they are going to form a replication bubble with replication forks at each end, where the parental strands of DNA are being unwound. And that replication bubble, it elongates as the DNA is replicated and eventually it fuses again. Since DNA is normally in a double helix shape, like a twisted ladder, before it can be copied, it has to be unwound and it has to be opened up. And this process involves a team of enzymes that work together. We have helicase. Helicase is an enzyme that's going to act kind of like a pair of molecular scissors. It's going to unzip DNA by breaking hydrogen bonds between base pairs, and then it ends up forming this Y structure, which is called the replication fork. We have single strand binding proteins, is what we see here. And what they do is that essentially once we have DNA strands that are separated, naturally they want to stick back together. So these single strand binding proteins, they come in and hold these two strands apart so that they stay open long enough in order for it to be copied. And after this, we have topoisomerase. So if you think about it, topoisomerase, it's like a helper that's going to relieve the tension that we have ahead of the unzipping area. When DNA is tightly coiled and when you start to unzip it, it can get twisted. What topoisomerase does is it holds together that DNA and it relieves the strain. So the key idea from this is that DNA can't be copied while it's closed. It has to be opened and it has to be stabilized first. So once we open the DNA, the cell is going to use each of the original strands as a template to build a new complementary strand. So DNA, it cannot be synthesized without a template strand. And at different parts of synthesis, the template strand that is being used is going to change. So a template strand, it's kind of like a guide or a pattern. Our cell, it's going to read each base on the original strand and add the matching bases. Remember, adenine pairs with thymine and guanine pairs with cytosine. This is going to make sure that the new DNA is going to be an exact copy of the original. Since both of the original strands are templates, the cell is going to end up with 2 new strands, 1 for each. And this matters because this is how that cell is going to keep the genetic information exactly the same every time it divides. So even though our original strands are ready, DNA polymerase, which is the main enzyme that builds new DNA, it cannot just start working out of nowhere. It needs this small section of nucleotides that are already in place to grab onto, and that's where primase comes in. Primase is an enzyme that creates a short RNA primer, and a primer is a short strand of RNA. And it's just a few bases long. So this primer, it gives DNA polymerase something to hold onto. And this matters because DNA polymerase, it cannot work alone. Primase is going to set the stage so that building can begin. And the way that primase does that is by having a free 3' OH group that our DNA polymerase can hold onto. So now that we have a primer in place, DNA polymerase can start to do its job. It's going to add new DNA bases. It's going to add new nucleotides one by one to the 3' end of the primer, which is right here. The new strand, it's going to grow in this 5' to 3' direction because the DNA polymerase, it can only read that way. So DNA polymerase will always build in the 5' to 3' direction. And what that means is that the new nucleotides are going to be added to that 3' end. So DNA polymerase, it uses the original strand as a guide just to make sure that the bases are paired correctly. And this is the actual copying part of DNA synthesis. It's how that new strand is formed. So with DNA, its strands, it runs in opposite directions. Kind of like 2 lanes on a street when they're going opposite way. One strand is going to run 5' to 3', and the other is going to run 3' to 5'. DNA polymerase can only run 5' to 3'. It can only build 5' to 3'. So both strands cannot be built the same way. And this is where things get a little bit more complicated. This is where we start to see a difference between the new two strands. And this matters because the direction of each strand affects how each new DNA strand is made. So this brings us to the idea that we have a leading strand and a lagging strand. So on one side of our unwound DNA molecule is the leading strand. The leading strand is where DNA polymerase has an easy job, right? So the leading strand is being continuously built. It is going to follow that replication fork, which is the place where DNA is being unzipped. It's going to build the new strand in the same direction as the fork is opening. So the fork is opening this way. So that it's being built this way. And when we are talking about the leading strand, we only need one primer at the very beginning, which is this little darker blue section. So DNA polymerase, basically, it just adds bases smoothly without stopping. And this strand is built really efficiently and really quickly. So now that we talked about the leading strand, we also have the lagging strand. So because DNA polymerase, it can't follow the replication fork because it builds in the opposite direction. Instead, it works away from the fork, building in small sections called Okazaki fragments. Every single one of these Okazaki fragments, it requires its own primer. And remember that RNA primer is made by primase. So after this, DNA polymerase is going to build short stretches of DNA until it bumps into the previous fragment. And it's important because the lagging strand, it's built in pieces, and then later those pieces have to be connected. So after all of the fragments are built, DNA needs to be cleaned and it needs to be connected. The first step is that RNA primers have to be removed because DNA should only be made of DNA, not RNA. So DNA polymerase, it's going to fill in the gaps with DNA bases, the gaps that we have from where these RNA primers were removed. Then we have an enzyme called DNA ligase that is going to come in and it's going to seal the gaps in between our Okazaki fragments. So ligase, it acts like a glue that is going to make sure that that lagging strand becomes one continuous piece of DNA. So DNA polymerase, it not only is like the builder, but it's also a proofreader. As DNA polymerase adds new nucleotides, the growing strand is going to check each one to ensure that it matches a template strand. If there is a mistake, for example, if an adenine pairs with a cytosine instead of a thymine, DNA polymerase is going to backtrack and it's going to remove that incorrect base and add the correct one. And that is known as proofreading. That is what I mean by DNA polymerase has exonuclease activity. It can literally remove an incorrect base if it needs to. Sometimes, however, the mistake can slip through, especially after replication, and that's where mismatch repair comes in. In mismatch repair, we have a team of enzymes that is going to find and correct errors. It's going to find and correct the errors that DNA polymerase missed. The enzymes, they look for mismatched pairs, and that newly made DNA, they're going to cut out that incorrect part via nucleases, and then we're going to have another enzyme fill in the correct bases. And then that final strand is going to be sealed by DNA ligase. So if we did not have mismatch repair, which mismatch repair is really essential for preventing mutations. Mismatch repair, it helps maintain the integrity of the genetic code, and without it, errors would accumulate over time and potentially lead to disease. So accurate replication and repair of DNA damage, they are important for the functioning of the organism and for passing on a complete and accurate genome to the next generation. And although the error rate is really low, rare mistakes such as mutations, they do slip through. Mutations, they have the ability to change the phenotype of an organism. If a mutation occurs in a germ cell, and remember germ cells, they give rise to gametes, those mutations, they can be passed on from generation to generation. And most of these mutations, they are harmful, but a really small amount of them can be beneficial. So mutations, they are that source of variation of which natural selection operates during evolution, and they are ultimately responsible for the appearance of new species. So limitations of DNA polymerase exist. DNA polymerase, it creates problem for linear DNA of our eukaryotic chromosomes. And this is because DNA polymerase has no way to complete the 5' end of the daughter strands, the DNA daughter strands. And every single time that we have replication, we're actually producing shorter and shorter DNA molecules. The ends of eukaryotic chromosomal DNA molecules, they have this special nucleotide sequence that is called a telomere. So telomeres, they do not contain genes. Instead, the DNA, it typically consists of multiple repetitions of a short nucleotide sequence. Telomeric DNA, it acts as a kind of buffer zone that is going to prevent the organism's genes from getting harmed. So it's a buffer zone that protects that organism's genes. Telomeric DNA and specific proteins associated with it prevent the staggered ends of the daughter molecule from activating the cell system for monitoring DNA damage. These telomeres, they become shorter after every single round of replication, and it might be thought that this is why we have aging. And it's also been suggested that the shortening of telomeres is connected to not only aging of certain tissues, but just aging in general. So eukaryotes, they have evolved a mechanism to restore shortened telomeres in germ cells only. So we can only elongate telomeres in germ cells, not in somatic cells. And the way that this happens is that there's an enzyme called telomerase. Telomerase is going to catalyze the lengthening of telomeres in eukaryotic germ cells. We do not have telomerase in the somatic cells, and the normal shortening of telomeres, it might protect organisms from cancer because it limits the number of divisions that somatic cells can undergo. But active telomerase, it's actually been found in some cancerous somatic cells. So most bacteria, they have a single circular double-stranded DNA molecule associated with a small amount of protein. A bacterium has a dense region of DNA called the nucleoid. Inside of the nucleoid, proteins cause the chromosomes to tightly coil, so they supercoil densely so that it fills only a small part of the cell. In the cell, eukaryotic DNA is packaged with protein to form chromatin. So DNA associated with protein is chromatin. The packaging of chromatin, it varies during the course of the cell cycle. So when we have condensed chromatin, it's called heterochromatin. But when we have dispersed chromatin, it's called euchromatin. Because of its compaction, heterochromatic DNA is normally largely inaccessible to the machinery in the cell that is responsible for expressing the genetic information that is coded in the DNA. More loosely packed euchromatin makes its DNA accessible to this machinery, so the genes present in euchromatin, the genes that are present in euchromatin, can be expressed. So remember that the chromosome it's a dynamic structure that is condensed, loosened, and modified, and then remodeled as necessary for a variety of cell processes such as mitosis, meiosis, and gene expression.